Welcome back. In the last web lecture, we were thinking about sexual life cycles that involve this process of meiosis or reduction division to create gametes with haploid numbers of chromosomes, and how this process of meiosis made possible a huge amount of genetic variability within populations. And we introduced this by talking about how important that variability is to the process of evolution by natural selection. Remember, evolution is the change over time in heritable traits. And so one of the major observations that Darwin made is that individuals in a population vary in their traits, and so we were considering how that variation comes about. And then the second part of this is that many of those traits are heritable. They're passed from parent to offspring. So in the next couple of web lectures, we are going to think about what are the rules that govern that passing on of traits from parent to offspring. How does this happen? And so remember that at the time Darwin was formulating this model of evolution by natural selection, he knew nothing about how inheritance took place. So in Darwin's time, this was considered a very, very important question, how this property of heritability actually works. So now we can think of this as drawing from the deck of genes, but of course genes were completely unknown at that time. So what we want to know is what principles account for the transmission of traits from parents to offspring. And in Darwin's time, really the best idea they had to work with was something called the blending hypothesis. So this is the idea that genetic material, whatever that turned out to be, it came from the two parents and then it just sort of mixed together, sort of like paints will mix together. So for example, like this blue and yellow paint blending together to make green. And Darwin himself found this explanation fairly unsatisfying. He thought that there were several observations of heredity that didn't really support this idea, but it was literally the only idea that they had at the time. It made the most sense at the time. So an alternative hypothesis that we know about now is something called the particulate hypothesis. And so this is the idea that parents pass on discrete heritable units that remain intact, and we call these genes. So as opposed to this blending idea where you mix blue and yellow paints and get green, you can have combinations of different characters, but the original parental characters are still there and can reappear in future generations. And so the main distinction between these two hypotheses, the blending and the particulate hypothesis, is that with blending inheritance, the parental types can never be fully recovered. So it's possible to have, if you're mating blue and yellow, it's possible to mate blue and yellow one time and then have that green offspring mate with yellow and then more and more yellow for generations and get something that's mostly yellow. That blue is always going to be in there. And so that's sort of the nature of this blending idea, is that those parental types completely disappear in their pure form once they're blended with something else. With the particulate inheritance hypothesis, the parental types are kept intact. So these are the two sort of major predictions that are made by these two different hypotheses. Either those parental types can be recovered in future generations, or they really can't. So one of the things Mendel did was documented a particulate mechanism, and he did this through experiments with garden peas. So first we're going to think a little bit about the approach Mendel used, how he used the scientific method to identify two of the very most important laws of inheritance. So Mendel discovered these basic principles of heredity by breeding garden peas in very carefully, very controlled experiments. So let's take a look at his approach. So Mendel's approach allowed him to deduce principles that had remained elusive to others. As we know, genetics, inheritance can be incredibly complicated. There are lots of factors involved, and to be able to really zero in on these fundamental principles requires a very meticulous approach and very carefully controlled experiments on very simple traits. So a heritable feature that varies among individuals, such as flower color, is called called a character, and each variant for a character, such as purple or white color for flowers, is called a trait. So the character would be color, and say, for example, purple would be the trait for that character. Garden peas were available to Mendel in many different varieties. He had a lot of variability to work with in his experiments. So peas, in this case, were used as a model organism. So what do we mean when we say a model organism? Model organisms have several characteristics. First of all, they are species that are used for research, and they're chosen because they're very practical to work with. Sometimes it's because they have a very short generation time. Sometimes it's because they are easy to breed in a laboratory. For whatever reasons, they're very tractable systems. And for a model organism, the conclusions that you draw from them need to be able to be expanded and broadened to apply to other species. So for genetics, for example, some common model organisms are for animals, Drosophila melanogaster, the fruit fly, is often used as a model organism because of its very small genome size and because it has many traits that are very clearly distinguishable from each other. In the plant world, Arabidopsis thaliana is a common genetic model system, again, also because of a very small genome size and very short life cycle. It's very easy to breed in a greenhouse. But for Mendel, what he had available as his very simple model system was the garden pea. So he chose these peas because, first of all, they're plentifully abundant, inexpensive, very easy to grow in his monastery garden. They have a very short generation time, so they're going to grow fast and you'll get results in a reasonable amount of time. They produce a very large number of seeds, so you can have very large sample sizes for experiments. And it was easy for him to control which parents were involved in a mating, and we'll take a look at exactly how he did that in a little bit. But also they have several polymorphic traits, so traits that can come in different forms. So a trait that appears commonly in 2 or more different forms, for example, purple versus white flowers. Mendel chose to track only those characters that occurred in just 2 very distinct alternative forms that you didn't see in your population of plants anything in between. It's either this or that. That made it very easy to score whether a trait is present or absent. So now let's take a look at the particular traits Mendel used. So Mendel worked with pea varieties that differed in 7 easily recognizable traits: seed shape, seed color, pod shape, pod color, and flower and pod position, and then finally stem length. An individual's observable features comprise its phenotype. So this is the outward expression of what we would call now its genotype, or the genes that it possesses. This is what the organism looks like. So we call that the phenotype. And Mendel's pea population had 2 distinct phenotypes for each of these 7 traits. So for seed shape, we had either round or wrinkled, and they're very easy to tell them apart. There's nothing in between. They could be yellow or green in color. The pod could be either sort of puffy and inflated or constricted so that you can see sort of the shapes of the peas inside the pod. The pod color could be either green or yellow. The flower could be purple or white, and it could be growing either at the ends of branches or at the very tip of the plant, so axial or terminal flower location. And these pea plants could be either tall or short. They didn't have this continuous variation between the height of the stem. Mendel's first experiments involved crossing pure lines that differed in just 1 trait. So in other words, he would have 2 plants that one had purple flowers and the other one had white flowers, but in all of these other traits they were the same. They either all had, let's say, round yellow seeds with inflated green pods growing axially and were tall plants. All of those things would be identical between the 2 groups. The only thing that would differ between them is this 1 trait that he was looking at. And this kind of approach of mating parents with 2 different phenotypes for a single trait and are otherwise similar is called a monohybrid cross. Mono being 1, so they're going to be hybridized for just 1 feature. So let's take a look at how pea plants normally reproduce. Pea plants normally self-fertilize. So the individual flower will produce pollen from the stamen, and the pollen from the same flower is going to be deposited on the female organ to fertilize those eggs. So they're generally self-fertilizing. So what he did was he started with varieties that were true breeding, which means that you can breed them generation after generation, allowing them to self-pollinate. They're always going to produce the same variety. So if it's purple, you can let it keep self-pollinating generation after generation. It will always produce purple flowers. So we call this true breeding varieties. So in a typical experiment, Mendel mated two contrasting true breeding varieties to each other in a process called hybridization. So let's take a look at how he did that, given that these plants typically self-pollinate. So what he did was, from some of the plants, he removed the stamen, these organs that produce the pollen, before the pollen developed. So there would be no opportunity for this plant to fertilize itself. Then he would take pollen grains from a different plant that he wanted to breed to that plant on a paintbrush or some other instrument and actually apply that pollen to the flower, cover them up so that no other pollen can access that flower, so you know who both of the parents of the next generation are. You can carefully control which plants are mating together. So a little bit of terminology that you need to know about. So these true breeding plants that we begin with are called the P generation. P is for parental. So these are going to be the pure breeding tall plants for this example, and so that's represented by having a tall self-fertilizing to produce another tall plant. So this is true breeding. A true breeding short plant, you've got one generation self-fertilizing to produce another generation of short ones. We know that these are true breeding, so this is going to be our P generation or parental generation. So step one is making sure you've got a true breeding line. Then you create hybrid offspring. And the hybrid offspring of the P generation are called the F1 generation. So you fertilize one true breeding type with a contrasting true breeding type in a carefully controlled way to produce the F1 generation that are hybrids between these two true breeding types. Then you either cross members of this F1 generation or you just allow them to self-pollinate. And the offspring from those pollinations are going to produce the F2 generation. So F, in the case of both F1, F2, is for filial. So think of that as an offspring generation. So we've got F1 generation, the first filial generation, and F2, the second filial generation. And then this F2 is going to produce some combination of traits. So by using this procedure, Mendel formulated what he called the law of segregation. So let's take a look at what results led into this and what exactly the law of segregation is. So we start with this P generation. We're going to use flower color in our example, using true breeding purple and true breeding white flowers. When Mendel crossed contrasting true breeding white and purple flowered pea plants, all of the F1 hybrids were purple. Every single one of them, and the purple was indistinguishable from the purple of the parental generation. If he had stopped there, we wouldn't really have learned very much. We don't know if this white has been completely obliterated, if it somehow mixed in with the purple and just can't be detected. We don't know what happened to this white based on just this one generation. Where we really start to discover things is when we do this next cross. And so when Mendel crossed the F1 hybrids, many of the F2 plants had purple flowers, but some of them had white, and the white was pure white, identical to the parental generation. So now we know this white factor was not lost. It was still there waiting to come out in a future generation. So this is consistent with the particulate hypothesis of inheritance as opposed to the blending hypothesis. And when he sort of crunched the numbers and counted all the flowers, what he found was that in this F2 generation, there were roughly 3 purple flowers for every 1 flower. So a ratio of 3 to 1 in the F2 generation. From these results, Mendel reasoned that only the purple flower factor was affecting flower color in the F1 hybrids. For some reason, whatever heritable factor was causing this to be purple was the only one having an influence. We know that the white factor had to be there because this parent only had white flower factor to contribute, but for some reason it's not being expressed or seen in this F1 generation. So Mendel called the purple flower color a dominant trait and the white flower color a recessive trait. So recessive implies hidden. It's not being seen. We know it's got to be there because it had a white parent that only had the white factor. So the factor for white flowers was not diluted or destroyed. We know this because it came back in full strength in the F2 generation. And this was what Mendel observed for all of these 7 different traits that he looked at. The same pattern of inheritance, the same ratio of offspring in the F2 generation. So what Mendel was calling a heritable factor is what we now call a gene. And Mendel was trained in mathematics and was looking for patterns, so he developed a hypothesis to explain that 3-to-1 inheritance pattern he observed in F2 offspring. And 4 related concepts make up this model. We'll go over them one at a time. And we also now can take these 4 concepts and relate them to what we now know about genes and chromosomes. And so we're going to first show you what Mendel observed and then explain them in terms of what we now know in terms of genes and chromosomes. So let's look at the first observation, the first deduction. So the first component of this model is that alternative versions of genes account for variations in inherited characters. For example, the gene for flower color in pea plants exists in 2 different versions, one that causes purple flowers and one that causes white flowers. So we call these alternative versions of the same gene, we call them alleles. So there's a purple allele and a white allele for the flower color. And so what we now know is that each gene resides at a specific locus on a specific chromosome. So here we have 2 homologous chromosomes showing the same homologous stretch of DNA at the same spot on that chromosome. So this particular individual has 1 chromosome that codes for a functional enzyme that allows the synthesis of purple pigment. This other one is a mutated form, a different version of that with some differences in the nucleotide sequence. So this one has an A where this one has a C. That mistake, that mutation results in a non-functional form of that enzyme or the inability to produce that enzyme so that the purple pigment is not produced. In the case of dominant traits, 1 allele results in enough of the protein product from that gene to produce the full purple color. So 1 allele results in enough pigment to make that flower indistinguishable from a flower from a plant that has 2 copies of this functional gene. Second component of this model is that for each character, an organism inherits 2 alleles, 1 from each parent. So Mendel made this deduction without knowing anything about chromosomes. Chromosomes had not yet been discovered, but what makes the math work out is having 2 alleles and having 1 of them come from each parent. So we now know that the 2 alleles at a particular locus may be identical, as in the true breeding plants of Mendel's P generation. So here we've got our 2 chromosomes with this equivalent locus on each of the homologs where the 2 alleles reside, and in this case, this is one of the true breeding varieties. It's going to have a wrinkled allele on both chromosomes. Or the 2 alleles at a locus may differ, as in the F1 hybrids. And we know that the F1 hybrids must have had 1 of each allele because they could have only gotten 1 allele from one parent and they could have only gotten the other one from the other. So we know that it has both. So if we look at this particular spot on the chromosome where this seed shape gene is located, we have a wrinkled allele for that gene on one of the chromosomes and a round allele on the other chromosome. The third component of this model is that if the 2 alleles at a locus differ, then one, the one that he referred to as the dominant allele, determines the organism's appearance, and the other, the recessive allele, has no observable effect on the appearance. So we've got 1 allele that's going to be masked or hidden by the other allele. So in the case of our flower color, that allele was the purple flower color. So in our flower color example, the F1 plants had purple flowers because the allele for that trait is the dominant allele. So the fourth component of this model is really what the law of segregation is all about. So the 2 alleles for a heritable character separate during gamete formation and end up in different gametes. So you're going to get one of the two possible alleles from each of your parents. Thus, an egg or a sperm gets only one of the two alleles that are present in the parent organism. And this segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis, what we saw in the last web lecture. So duplication leads to two pairs of sister chromatids for the two homologous chromosomes. They're going to separate during meiosis 1, and then those two sister chromatids are going to separate again, and only one of those two versions of that gene, only one of those two alleles is going to end up in each one of the gametes. So this model accounts for that 3-to-1 ratio observed in the F2 generation of Mendel's crosses. This is the reason he was able to come to this conclusion, because this is the solution that gives you that 3-to-1 ratio. So let's take a look at how. So the possible combinations of sperm and egg can be shown using a figure called a Punnett square. And so we'll build up to exactly what that is. So if we start with our cross of true breeding parentals, so each of these true breeding purple flowers has two copies of the purple allele. And so a capital letter usually is used to represent a dominant allele, and the same letter in lowercase generally represents the recessive allele. So we know that these true breeding purple flowers are big P, big P. So they are always going to contribute a big P when they create their gametes. That's all they have to give. The white flowers we know are little p, so all of their gametes are going to have a little p. That's the only allele they have. So when we look at the F1 generation with all purple flowers, we know that each one of these got a big P from one parent and a little p from the other parent. They are all big P, little p combinations, and they're all going to be purple because the purple allele is the dominant allele. So every one of them is purple. So when these F1 plants create gametes, they've got a big P and a little p, and they're going to put them into their gametes in equal ratios. So every gamete has an equal probability of being either big P or little p. So half of their gametes will be big P, half of their gametes will be little p. So when you interbreed these individuals from the F1 generation with each other or with themselves, each one of them can contribute a big P or a little p. So if we talk about the sperm or the paternal contribution from the F1 plant, their gametes can either be big P or little p. So we're going to draw those across the top of this Punnett square, this box. And then along the side, we're going to list the possible gametes from the maternal contribution from the eggs, and that can also be either big P or little p. And so for each of these cells in the Punnett square, we're just going to look at what the paternal and the maternal contribution was. So in this square, it's big P, big P. This one got a little p from the father, big P from the mother, so it's big P, little p. This one got a big P from the father and a little p from the mother. So again, it's going to be big P, little p. And this one got a little p from the father and a little p from the mother. So if we look at the phenotype, the physical appearance of these F2s, 3 of them are going to have the purple phenotype, even though they have different combinations of alleles. And one of them is going to have that white phenotype, giving you that 3-to-1 ratio. So let's talk about some of the vocabulary that we use. So an organism with 2 identical alleles for a character is called a homozygote, and it is said to be homozygous for the gene controlling that character. This is a purple homozygote, this is a white homozygote. An organism with 2 different alleles for a gene is a heterozygote and is said to be heterozygous for the gene controlling that character. So here, middle, we've got purple heterozygotes. They are heterozygous for the flower color character. So unlike homozygotes, heterozygotes are not true breeding. So if you take these heterozygotes and allow them to self-fertilize, they're not going to produce all purple flowers because they've got these white alleles in them. They're going to produce that 3-to-1 ratio. So the important thing to note is that an organism's traits do not always reveal its genetic composition. So if all you saw was purple flowers, you would have no way of knowing whether any of these individuals was homozygous or heterozygous. So we've got 3 different ways to become purple that result from basically 3 different combinations of gametes. Either homozygous, getting a big P from both parents, you can get a big P from the mother and a little p from the father, or a big P from the father and a little p from the mother to make a heterozygote. And then, of course, you always know the genetic makeup of the recessive phenotype because it's always going to be homozygous recessive. So this is the one case where you can infer the allele composition based on the phenotype. So we've got a phenotypic ratio of 3 to 1, so 3 purple flowers to every white flower. If we don't really care which one of these alleles came from the mother and which one came from the father, we can say that these heterozygotes are equivalent. We're going to have a 1 to 2 to 1 ratio of different genotypes, either the homozygous dominant, heterozygous, or homozygous recessive. So we've got a phenotypic ratio reflecting the outward appearance, and we've got a genotypic ratio reflecting the composition of alleles leading to that phenotype. So therefore, we distinguish between an organism's phenotype, or its physical appearance, and its genotype, or its genetic makeup, the combination of alleles that it has. So again, in the example of flower color in pea plants, big P, and big P, little p plants have the same phenotype, but different genotypes. So if you have an individual with a dominant phenotype, how can you find out what its genotype is? It could be either homozygous, dominant, or heterozygous. How can you find out? So to do this, we use a technique called the test cross, and it's very simple. To determine the genotype, all we have to do is breed that mystery individual with the dominant phenotype with an individual with the recessive phenotype, because you know the genotype of that individual. It has to be homozygous recessive to be able to express that recessive phenotype. So here's what it looks like. So we've got a dominant phenotype of unknown genotype, and you cross it with your homozygous recessive type, in this case the white flower. And so there are 2 possible outcomes. If this is a homozygous dominant individual, you will get, from this test cross, all purple flowers, and you can use the Punnett square to verify that. We're hypothesizing that this is big P homozygote. We know that the white flower is little p homozygote, and so you get big P, little p, big P, little p, big P, little p, big P, little p, and all of the offspring you expect only purple flowers. If it's a heterozygote, you can again use the Punnett square to make a prediction of what the offspring should look like. So if it's a heterozygote, it's got a big P and a little p, so you're going to put that along one side. The little p, from what we know is our recessive homozygote, and so when you combine the big P and little p, you get purple. Big P and little p, you get purple. But then there's going to be little p with a little p to get white, little p with little p. So if this mystery individual was a heterozygote, you expect half and half purple and white flowers. So if any of the offspring display the recessive phenotype, you know that you must have a heterozygote. The other of Mendel's important genetic laws that he discovered is called the law of independent assortment. So Mendel derived the law of segregation by following a single character. And so the F1 offspring produced in this cross were called monohybrids. So the F1, remember, were all heterozygotes for that one character that he had isolated. Mono means one, so they're going to be a hybrid for one single character. So a cross between such heterozygotes is called a monohybrid cross. So Mendel identified his second law of inheritance, the law of independent assortment, by following 2 characters at the same time. He did this by crossing 2 true breeding parents differing in 2 characters rather than just one, with all of the other characters being similar between the 2 groups, to produce dihybrids, so hybrids for 2 characters in the F1 generation that are heterozygous for both of those 2 characters. So a dihybrid cross, a cross between F1 dihybrids, can determine whether the 2 characters are transmitted to offspring as a package, so always coming together, or are they inherited independently? Can they be separated from each other? So here's the question. Do different traits vary together? Are they inherited together, or are they inherited independently of one another? And so, for example, if you are looking at yellow and round individuals, is that yellow trait and that round trait kept together in the gametes, or are they separated in the gametes? These two hypotheses, either dependent or independent hypotheses, make very different predictions about the phenotypic ratios that you would expect in the F2 of a dihybrid cross. So let's take a look at how that works. What we're going to do is we're going to take our parental generation of true breeding plants with yellow round seeds and cross them with parental true breeding green wrinkled seeds. All of these gametes are going to be big Y, big R. All of these are going to be little y, little r. Those are the only gametes that each of these have to offer. And so all of the F1 generation are going to be big Y, big R, little r. So the question is the big Y going to always stay with the big R, and is the little y always going to stay with the little r? If it does, then you can only have 4 different kinds of gametes. Big Y, big R, little y, little r, for both the paternal and the maternal factors. So this gives you a very simple Punnett square where you recover those parental types in a 3-to-1 ratio. The double homozygotes, the double heterozygotes, and then the double recessive homozygotes in a 3-to-1 ratio. So this is the hypothesis of dependent assortment, where those 2 alleles stay together and are inherited as a unit. The alternative hypothesis is the hypothesis of independent assortment, so that big Y can be inherited either with big R or with little r, and little y can be inherited either with big R or little r. So that gives you 16 different possible combinations for the F2 offspring. So you can, for the sperm, each of the gametes can either have big Y, big R, big Y, little r, little y, big R, or little y, little r. Same for the eggs, those 4 possibilities. And so they can combine in 16 different ways. So the big difference here is in this situation, you can get yellow and wrinkled, which you can't get here in this dependent scenario. And you can get green and round. Those are both possibilities if they assort independently. So in this case, you would expect a phenotypic ratio of 9 of the dominant phenotype, both yellow and round, 3 for the dominant round shape and the recessive green color. So you can just count these off in the different cells showing the results. You'd expect 3 of the dominant color, yellow, with the recessive shape, wrinkled. And so again, you can just count how many cells that appears in to get that. And then just 1 of the double recessive, green and wrinkled. So it's going to be a phenotypic ratio of 9 to 3 to 3 to 1 for the dihybrid cross if these traits assort independently from each other. So let's take a look at what actually happened. So it turned out that we did get combinations of the dominant and recessive phenotypes. So we did get green round and yellow wrinkled, and we got them in roughly the phenotypic ratio of 9 to 3 to 3 to 1. So this shows that those 2 traits are inherited independently from each other and can combine in any possible combination. Using a dihybrid cross, Mendel developed the law of independent assortment. So it states that each pair of alleles segregates independently of any other pair of alleles during gamete formation. So not only do the 2 alleles separate from each other, they do this completely independently of the other alleles at the other genes. So this applies only to genes on different non-homologous chromosomes or those that are far away from each other on the same chromosome. We know this now. Mendel didn't know this at the time. So we know now that chromosomes are the actual unit that is passed down. So why do they need to be far away on the same chromosome? So genes located near each other on the same chromosome do tend to be inherited together and show patterns of dependent inheritance. So what's the difference? So it comes back to this idea of crossing over during meiosis. because genes that are farther away from each other are more likely to have a crossover event in between them that are going to separate those two genes from each other than genes that are very close together. Genes that are far apart on the same chromosome are more likely to behave in this independently assorted way because they can be separated from their associated gene on the same chromosome relatively easily by crossing over. So genes that are far away from each other on the same chromosome are going to have this independent assortment behavior just as if they were located on two completely different chromosomes. So we've seen a little bit of how these combinations occur at random. And so what this means is that this whole process is going to be described by laws of probability. So laws of probability apply to randomly generated results. Mendel's laws of segregation and independent assortment reflect the rules of probability because they're random processes. So, for example, when tossing a coin, the outcome of one toss has no impact on the outcome of the next toss. Each one is a completely separate and completely random event. So in the same way, the alleles of one gene segregate into gametes independently of another gene's alleles. So remember we said when we looked at meiosis that these homologous chromosomes can line up in any combination of maternal and paternal. And imagine instead of 2, we've got 23 that are all aligning at random along this metaphase plate. And so you've got a completely random assortment. And then lay on top of that the random possibility of crossing over. You've got a completely random process that is going to be subject to the laws of probability. So what are these laws of probability and how do you calculate the probabilities of these events happening? So I like to think of the multiplication rule as the and rule. So if you're putting this into words and into a sentence, this is going to be the rule that determines whether two separate events, this and that, both happen. Okay, so the probability that two or more independent events will occur together, and you're going to get that probability of both of those things happening by multiplying, getting the product of their individual probabilities. The probability of getting a homozygote dominant is going to be equal to the probability of getting a big R from the sperm. That probability is 1/2. It can be either big R or little r with equal probability. And the probability of getting a big R from the mother, which again is a probability of 1/2. So the probability of having a dominant homozygote big R is the probability of this, and the probability of that, multiply them together, 1/2 times 1/2 is equal to 1/4. And each of these combinations has a probability of 1/4 because getting each of these gametes along each of these columns or rows is going to be 1/2, and you just multiply them together. So the probability in an F1 monohybrid cross can be determined using that multiplication rule, what I like to call the and rule. Segregation in a heterozygous plant is like flipping a coin. Each gamete has a 1/2 chance of carrying the dominant allele and a 1/2 chance of carrying the recessive allele. So now let's look at the addition rule, and I like to think of this as the or rule. So this is the probability of this or that happening. So if there are multiple ways to get a particular outcome, what's the probability of getting this or that? So the addition rule states that the probability that any one of two or more mutually exclusive events will occur is calculated by adding together their individual probabilities. So the rule of addition can be used to figure out the probability that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. So let's take a look at how this works. The probability, so I'm using lowercase p to stand for the probability of getting a heterozygote, you can do it in one of two ways. You can get the dominant allele from the mother and the recessive allele from the father. So it's going to be the probability of dominant from mother, probability of recessive from father. So this and that has to happen. Or you can get the recessive allele from the mother and the dominant allele from the father. So the probability of getting the recessive allele from the mother and getting the dominant allele from the father. So you have to get this and that to get a heterozygote in this way, one from the mother, the other one from the father, or recessive from the mother, and dominant from the father. There are two different ways of getting a heterozygote. So how are we going to calculate this overall probability? So the probability of being heterozygote because you got dominant from mother and recessive from father is 1/2 times 1/2, or 1/4. Okay, that's dominant from mother, recessive from father is right here. So you multiplied these two probabilities to get the probability of being this kind of heterozygote. Or you could also be a heterozygote in this way, getting the recessive from the father and the dominant from the mother, with a probability of 1/4, 1/2 times 1/2. So this is the probability of dominant from mother, recessive from father. This is the probability of recessive from mother and dominant from father. To know if you're going to get this result or that result, these are two different ways to be a heterozygote, you add them together. So the probability is 1/2. And you can see this very easily. You have 2 chances out of 4 of being a heterozygote, 1/2. So you can use these and or rules, these multiplication and addition rules, to solve much more complex genetics problems. There are detailed examples of this in your book. We're not going to worry about it too much for our purposes, but we just be sure that you understand that we can apply these rules of probability to predict the outcome of crosses involving any number of characters. So a multi-character cross is equivalent to 2 or more independent monohybrid crosses occurring simultaneously. That's, of course, assuming that those 2 genes are not right next to each other on a chromosome. So in calculating the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. So if you want to know the probability of getting all of these separate genotypes, this and that, you multiply together. If you want to know the chances for a result that can be obtained in multiple ways, for example, the example in your book was at least 2 traits that are homozygous recessive among whatever number of traits you're looking at, then you calculate all the different ways that you can get that outcome and then add together those probabilities. So getting this or that, you're going to add the probabilities together. So in this web lecture, we looked at the relatively simple cases of heredity that Mendel studied, but you may be saying to yourself at this point, you know, this is very nice and easy to understand, but it can't really be that simple. I know perfectly well that if I have a tall mother and a short father, I'm not going to be exactly tall like my mother or exactly short like my father. I can be anything in between, or even taller than my mother or even shorter than my father. It just isn't that simple. It's having 2 traits and you get one of them or the other. So in the next web lecture, we're going to look at some of these complexities that cause what we call continuous variation. And so that's what the graph you're looking at shows this. There's not just 2 states for height in human beings or most species. There's going to be a continuous range of variation in height. And so clearly the actual genetics involved is more complicated than just purple or white flowers or round or wrinkled peas. Mendel chose those characters because they were simple. So in the next web lecture, we're going to look at some of the factors underlying some of these more complex cases of inheritance involving environmental factors, the effects of multiple genes on a single trait, and other ways that this can become more complicated. We're also going to look at some of the ways we can use simple Mendelian genetics to understand human health and heritable diseases.